I'm going to move into the question set that has come through from the audience, because we have quite a few, and I want to make sure that we're addressing these questions. First one, where can we find exosome clinical trials in the US? And feel free to chime in.
Well, I'll start. Since Codiak BioScience is currently conducting a clinical trial with a compound we call Exosome. It's a phase 1/2 trial in immuno-oncology for treatment of injectable solid tumors. That study is ongoing, and Codiak is projecting a first readout of data within Q3 of this year. So very exciting. Codiak is also conducting another clinical trial that's outside of the United States in the UK with XORL12. That has already reported the first clinical trial data from a randomized control trial set in healthy volunteers, and are projected to report first data readout in patients with cutaneous T-cell lymphoma at the end of 2021. I think, other sources of clinical trials, I'm sure clinicaltrial.gov is a good place to look. I kind of know, the own backyard the best, but there's not many. Codiak has been the first to really advance engineered exosome therapeutics into the clinic. And I think Dr. Mari will be in a better position to kind of answer, what kind of clinical trials are taking place with exosomes just harvested from different cell lines and trying to leverage their potential endogenous kind of pharmacological properties. So I think she'll be in a better position to further that question.
Sure, Dr. Murray, go right ahead.
Yeah, thank you. So yeah, Organicell is actively recruiting for different trials. Clinicaltrials.gov will be the best way to find them. We have already launched our results of the expanded access for COVID patients that took place in Texas. And right now, waiting for the approval of phase 2 for the COVID mild to moderate. We think that it will be maintained in Texas, but other sites are opening up as well. Our osteoarthritis, I can't disclose yet the location itself, but it will be in New York City with other multiple sites, as it is a multi-center. Our COPD trial will be enrolling in Phoenix. Again, cannot disclose yet the site, but you will be able to find it in clinicaltrials.gov. And our long hauler trial, which is what we're most excited about right now, as it is a set of patients that, quote unquote, has been forgotten from this pandemic, but we will be announcing really soon where patients can enroll. Clinicaltrials.gov will be where you can find that information.
Okay, thanks, Dr. Murray. Next up, here's a question regarding specific cell sources. So what are your thoughts, and maybe Nicolas, we could start with you. What are your thoughts on picking specific cell sources such as MSCs for natively therapeutic extracellular vesicles rather than engineering EVs after they're isolated? Does further engineering add potentially disruptive complications?
Well, I'm not sure I fully get the question. Well, are we in the, well, it depends if the application is regenerative medicine. I guess so, if the question is on stem cells. My answer would be the traditional answer, probably of stem cell developer, is if you have primary cells, you have the challenge of having variability of donors, and you need to repeat cell banking once your source arrive to some passages because of senescence of your cells. So either you can use some sources that are more maybe less older cells from placenta or from umbilical cord. But otherwise, I think a good strategy is to try to immortalize cells or to use IPS. You can also, which is also interesting, I think not enough investigated in the literature by current companies to have some priming of your cells, trying to add some other component to give to your exosomes really some properties, interleukin. You can use lipids or other kind of components that I think can also affect the therapeutic efficacy of your product, and I think priming is an interesting strategy in the regenerative space of exosomes.
Okay, thanks, Nicolas. And just to make sure that we heard the question properly, I'm going to ask it again. What are your thoughts on picking specific cell sources, such as MSCs, for natively therapeutic EVs, rather than engineering EVs after they're isolated? Do you feel that further engineering adds potentially disruptive complications?
I can answer that question a little bit, Erin. I think, this is really a matter of whether you want to pursue a unique therapeutic hypothesis where you are assigning the pharmacological payload to the exosome, or whether you are trying to kind of follow some, endogenous kind of payloads that could generate different kind of pathway activations that could be biologically or pharmacologically interesting. It really is a matter of how much control do you want. We at Codiak BioScience recently published a paper that described how we are engineering and generating, if you will, bespoke exosome therapeutics, where we are assigning the pharmacological property. And, it certainly generates, a very, very clear therapeutic hypothesis. Either the exosome therapeutic is working or not, and you can measure and you can do PK and PD analysis. And I think that this is really the key question to ask. I have not seen or heard of that, assigning pharmacological properties to exosome is disruptive or could be dangerous. If anything, I think it generates a high level of predictability and potency that I believe is very important for further advancing the field.
And maybe just rapidly, because I think we are running out of time, but just to complete, you have either the strategy of engineering your cells or produce your EVs and engineer your EVs. And I think what Codiak is doing is mostly engineering the cells. And I think this is, the easiest strategy and more robust strategy to go on, as it's still difficult to engineer EVs post-production, if I am not mistaken, Benny.
Yeah, no, I can answer that quickly. That actually we are doing both. We are engineering cells to make exosomes that have pharmacological properties, and then post-purification, we can load on small molecules and oligonucleotides with a high degree of precision and potency.